Hello, welcome to GP Podcast. Today, we're diving into a true revolution, right at the intersection of AI and biology. Now, this comes from the same brilliant minds that gave us AlphaFold, but this time, they're not just looking at one piece of the puzzle. Oh no, they're trying to decode the entire instruction manual for human life.
Just think about that for a second. It's a pretty wild thought, right? But for decades, this has been the reality for geneticists. You see, our genome is this gigantic 3 billion letter-long code, but most of it doesn't actually create proteins, the building blocks of our bodies. It's been this huge mystery, a kind of biological dark matter. And today, we're going to explore the incredible AI that was built to finally shine a light on it. So, here's how we're going to break this all down. First up, we'll get a handle on just how big this genetic mystery really is. Then, we'll meet AlphaGenome, the AI designed to solve it. We're going to look at how it works, see how it stacks up against everything else out there, and most importantly, what its power could mean for the future of medicine. All right, let's get right into it. To really appreciate what a breakthrough this is, you first have to understand the sheer size of the challenge, this huge, uncharted territory that's inside every single one of our cells. So, have a look at this. Only a tiny 2% of our genome is made up of the protein-coding genes that we've traditionally focused on. The other 98%, well, that's this non-coding region. But here's the kicker, the really crucial part. This dark matter is where almost all the genetic variations that make us unique, and the ones that can lead to disease, actually live. For years, if a disease-linked mutation popped up in here, scientists were pretty much flying blind. This is the code that AlphaGenome was built to crack.
So, how in the world do you read the unreadable? Well, you build a totally revolutionary new reader. And that's where AlphaGenome comes in. It's an AI that doesn't just see the letters of our DNA, it's learning to understand the grammar, what the code is actually doing.
You probably remember AlphaFold. It was a total game changer for predicting protein shapes. Think of that like figuring out the function of one super vital component in a massive, complex machine. Well, AlphaGenome is the sequel, but its ambition is just breathtaking. It's designed to understand the entire machine, the full picture of which genes are switched on, how our DNA is all packaged up, and how all the different parts of our code talk to each other. So, how does it manage all of this? I mean, to pull off a feat like that, AlphaGenome needed a completely new design, a new architecture, a way of looking at our DNA that could capture both the tiny, microscopic details and the huge, panoramic view, all at the same time.
This right here, this analogy, it just perfectly captures the challenge. You see, older models had this trade-off. They could look at a little piece of DNA with really high precision or a really big piece with kind of blurry details. AlphaGenome does both. It reads a massive 1 million DNA letters at once, but it can still make predictions right down to a single letter. It's seeing the words, the sentences, and the entire plot of our genetic story all at once.
The secret sauce, the magic behind it, is something called multimodal training. Now, instead of creating a bunch of separate AIs for all these different tasks, like gene activity or DNA structure, they built one single unified system. It takes the raw DNA sequence and learns to predict a whole bunch of different biological outcomes at the same time, which lets it discover these deep shared rules that govern how our cells work.
And believe me, when I say a bunch of outcomes, I really mean it. This one single model predicts the results of what would normally take dozens of different lab experiments to figure out. We're talking about gene activity, which is what RNA-seq measures, DNA accessibility, or ATAC-seq, which is basically how open the code is to being read, all the way to the 3D structure of the genome itself. It predicts all of this from the exact same string of letters, replacing nearly 6,000 separate biological measurements. It's just incredible.
Now, as you can imagine, training a beast like this is a massive engineering challenge. It happens in two main stages. First, they train several large teacher models on tons of real data. Then, they use this really clever process called distillation, where a single, much more efficient student model basically learns all the combined wisdom from those teachers. And the result is a model that isn't just powerful, it's also lightning fast. It can analyze a genetic variant in under a second.
So, okay, the theory is really impressive, but how does it actually perform in the real world? Let's take a look at the data.
The performance is, well, it's frankly astounding. Just look at this chart. When they tested it on 24 standard prediction tasks, AlphaGenome was the top performer in 22 of them. This wasn't just a small improvement, it was an absolute domination. And this right here, this is where things get really, really exciting for medicine. Being able to predict the effect of a genetic mutation is kind of the holy grail in this field. And on 26 different tests that measure this ability, AlphaGenome matched or beat the best competing method on 25 of them. It is, to put it simply, the new state of the art.
Let's look at a specific example to really see the difference. EQ tails are basically genetic variants that act like dimmer switches for our genes, affecting how strongly they get turned on or off. As you can see in this table, the previous model had a correlation of about 0.39 with what actually happens. AlphaGenome boosted that to 0.49. Now that might not sound like a huge jump, but in complex biology, trust me, that is a major leap in accuracy. But what's even more telling is that bottom line. It found more than twice as many of these known dimmer switches as the next best model. It's just seeing things that others can't.
So what does all of this performance actually mean for our health? What happens when you point this powerful new lens at a real-world disease?
Let's walk through a fascinating case study that involves a blood cancer and a gene called tau1. In this case, the problem isn't a flaw in the gene itself, but in that mysterious non-coding DNA that's supposed to be controlling it.
So here's what's going on. You have these mutations in the non-coding region that create new on switches, which are called enhancers. These rogue switches then crank up the tau1 gene's activity to really dangerous levels, and that causes leukemia. And here's the truly mind-blowing part. AlphaGenome predicted this entire chain of events, the new switches, the out-of-control gene, the whole deadly mechanism, just from analyzing the raw DNA sequence.
This is the kind of deep insight into how a disease works that would normally take a team of researchers years and years of painstaking lab experiments to figure out. AlphaGenome saw the entire story written right there in the code itself. This means we can now potentially spot these cancer-driving mutations years before they cause any problems purely from a genetic sequence.
You know, this isn't just another small, incremental step forward. It really feels like a foundational shift, a brand new tool that moves us from just sequencing our DNA to truly, deeply understanding it. We're standing at the dawn of a new era in biology, one where the instruction manual for life is finally becoming readable. And that, of course, leaves us with a pretty profound question for the future.
If you liked the video, subscribe, leave a like, comment on what you liked most, and share the video.